Right, so I'm going to be giving a talk on isolating new bacteriophages for the purpose of treating antibiotic-resistant prosthetic joint infections. This is in collaboration with the University Hospital Coventry and with a great collaborator called Evangelos Veronis there, a really wonderful guy, really passionate about phage therapy, which is really nice to see in the sort of clinical sort of area. I'm so sorry I couldn't be with you guys today. I'm currently on placement with the Welsh Government for a water policy team, so it would have been hard for me to get there today. Anyway, so I'm going to give some context to all this, to this story. So, University Hospital Coventry, the infection have had some issues after surgery for giving prosthetic prosthetic joints and after giving prosthetic joints out, so such as hip replacements, knee replacements, they tend to get quite infected sometimes with organisms such as MRSA, E. coli, and basically these infections are hyper resistant to all of last line antibiotics for giving them. And another issue is because it's because it's under the skin, and it's very hard to actually give to treat these infections with the way to treat these prosthetic joint infections is by literally cutting it open, reopening the wound, and basically scrubbing down it's essentially a toothbrush to clear out these infections. And these infections are highly morbid, like causing almost amputations of limbs or even sepsis and death. So the hospital have looked at Evangelos has been looking into trying to treat these infections. Again, antibiotics don't work, so he's came to us, to Antonio's lab, to try see if we can isolate some new bacteriophages towards these specific organisms, these infections. And we received some clinical isolates from them, ranging from Staphylococcus epidermidis, Staphylococcus succinis, Warneri, capitis, and hopefully we can. And in this, we're going to try and see if we can use bacteriophages to treat these infections. So Staphylococcus, again, it's causing massive issues in hospitals with MRSA right now. Again, you all know it's all there's high amounts of antibiotic resistance amongst them, and in people such as older patients or immunosuppressed patients, it becomes an opportunistic infection. And in prosthetic joint infections, after orthopedic surgery, about 40% of infections after orthopedic surgery tend to be of Staphylococcus. So one of the solutions Evangelos has asked us to provide maybe is to look into phage therapy to see if we can clear these infections in these patients after obviously all the excitement around bacteriophages and their ability to clear biofilms recently. As again, these prosthetic joint infections, what's causing the issues is that they're forming like a thick biofilm on the prosthetics, which are very hard to get to clear. So one of the first phages we used, which is this is one we had in house for Staphylococcus. It's very well studied already. It's already been mentioned a few times in this conference. Phage K. We wanted to see if we could try using Phage K to clear these infections. Again, Phage K is mainly targeting Staphylococcus aureus, but we wanted to see if it but we've got clinical isolates varying from Staphylococcus epidermidis, aureus, Warneri, capitis. So we wanted to see if our Phage K could clear our other types of species of Staphylococcus. So initially, what we did was a quick clearance assay, again a lawn of bacteria, and then spot test 1 × 10⁸ PFU, like 10 microliters, 100 microliters of that, or 10 microliters, and onto a lawn of bacteria, see if there's any clearance. Phage, so this green is good clearance, orange is what we assess to be medium clearance, and red is no clearance. And for most of the organisms, our Phage K has no host range. So obviously at phage K isn't going to be very effective, from what we can see here. So what we try next is isolating new phages against these clinical isolates. Initially, we were going to try hospital water samples, but then I read around in literature and instantly found us. I mean, obviously, most of you probably know this methodology, but we found that sewage was to be the best place to find new phages. So we did that. We co-incubated filtered sewage with our Staphylococcus, all our clinical isolates at exponential phase, and that gave us lots of new bacteriophages, which we then isolated from each individual plaque, isolated and then tried to get growing. A lot of them were an absolute nightmare to get growing, but we managed to get a good 12 of them to grow quite well. We then sequenced by MinION and Illumina just to check that before we start thinking about applying these to patients, that there's no integrases or any other pathogenicity factors. And then we visualized by electron microscopy and just did some characterization. So we had our new library of phages against pathogenic Staphylococcus that we could potentially use for a phage cocktail. So we got 12 new phages, all of varying genuses. We found a couple of them. I think there was one 3.2. It's not listed here because we ended up kicking it out. But we found a couple of the ones we isolated to have integrases. So we kicked those out, and we ended up creating a new phage cocktail that we thought would have broad host range across all our different clinical isolates to make our new phage cocktail. And again, we use 4 phages along with phage K that also have found not to have any integrases or any other antibiotic resistance cassettes or anything. These are the electron microscopy images of them. Pretty. So how effective is our new phage cocktail in simply a spot test? We find that it's much more effective in clearing all the organisms compared to phage K. So there we go, we've already made an improvement there. But however, obviously a spot test is nothing like these infections are, these prosthetic joint infections are caused by biofilms, not by exponential phase bacteria. These are very established biofilms. So we wanted to try and recreate those conditions and to assess our phage K and phage cocktail against these clinical isolate biofilms, or prosthetic joint infection biofilms. What we did, or what I did was I got a glass cover slip, I plasma treated it to try and make it sticky, incubated it with our clinical isolates for 72 hours, shaking, so there was some shear stress, which should have strengthened the biofilm a bit. We then apply we want to compare against the antibiotics the hospital's currently using against them. So we apply our phage cocktail and our antibiotics, treat for 24 hours, and then we visualize these biofilms and we do live dead staining on these mature biofilms that have been treated. So again, we have our 3-day Staphylococcus biofilm. 1 day of treated. We then visualize it using confocal microscopy, and then we process in image J the proportion of live to dead cells. This is with propidium iodide / this live staining. And in image J, it gives us a specific proportion of living to dead cells. The dead cells will pick up propidium iodide, which will cause the cells to fluoresce red in confocal. So from doing this, we have from growing our mature biofilms and then treating them with one of the main last line antibiotics for treating for biofilms, we compared it with rifampicin at a normal concentration that's applied to the body. We use phage K on its own and phage cocktail about 1 × 10⁶. We find that's with rifampicin, it's not very effective in clearing the biofilms. Phage K, we find it to be more effective than rifampicin in clearing these biofilms, which is a good sign. And our phage cocktail, surprisingly, even though in the pure surface test, just when you spot test the phage, phage K or phage cocktail onto one bacteria, we found massive differences in its host range. In this case, we didn't find very significant difference in the host range between phage K and our phage cocktail, which was supposed to have a higher host range, which maybe describes that in mature biofilms, it may not matter. Like the host range of the phage may not be such a massive issue on its ability to clear the biofilm. So phages might actually have a broader host range for clearing biofilms than we actually thought, looking at some of the data I have so far. And another thing we noticed was after treat one of the clinical isolates that really produces quite thick biofilms, even when you're growing it on a plate, it produces this horrifying colony that's like spiky. Staphylococcus succinus. After vancomycin treatment or antibiotic treatment, we get increased biofilm formation compared to no treatment at all. So if anything, in these prosthetic joint infection patients, the antibiotic treatment might be causing more issues, might be inflaming the issue. However, with phage cocktail, we find much higher killing, almost complete killing in some cases. So considering that in the hospital, the phage cocktail will be being used alongside antibiotics, we wanted to assess how well will our phages new phages assist treatments in these prosthetic joint infection patients. And I just want to point you here to this figure here. So first, we use a minimum biofilm eradication MBEC assay. It's a very common assay where you use a textured peg, and then just on a 96-well plate, and the textured peg can really pick up, helps form the biofilm. Then you treat it for you form a biofilm, treat it for 12 to 24 hours. In your treatment, either whether it's antibiotic or antibiotic and phage cocktail. And we found that the minimum amount of antibiotic concentration required when we use our phage cocktail is reduced absolutely massively. Again, a 20 times decrease in almost all cases, which really signifies in vitro that there's a much higher chance of success in treating these patients if we use alongside bacteriophage therapy. And again, the biofilm mass with phage cocktail is massively reduced as well, which spells really good news for us. And one of the last things, before we even start thinking about giving our new phage cocktail to humans, we wanted to assess, is it toxic to humans? So to do this, we did an in vitro assay in human. So we picked human epithelial cells. We thought of this because that's we decided to choose that because that's the one we thought basically most appropriate to use T24 human epithelial cells. We exposed these epithelial cells to normal concentrations of rifampicin, flucloxacin, and vancomycin. Again, concentrations that you normally find in the body, and then compared it to and then also exposed T24 cells to our phage cocktail about 1 times 10⁶ PFU per mil, and we found no significant difference in the cytotoxicity to human cells compared to antibiotics. So phage cocktail compared to antibiotics is no more toxic. Even when used alongside antibiotics, we didn't see very much more increase in cytotoxicity. How this is assessed, cytotoxicity, is that if the cells, again, once you've exposed the cells to your treatment, depending if the cell, depending on the amount of cells that have died and lysed in the culture, that will release lactate dehydrogenase, which will cause enzymatic change in the color of the solution, which you then can check on a 96-well plate reader, and then you can see the general percentage toxicity compared to controls. So in summary, we've isolated and characterized some new bacteriophages against various species of Staphylococcus, again, Epidermidis, Waneri, Capitis, and Staphylococcus aureus. We found that phage therapy is more effective than antibiotics in clearing biofilms of Staphylococcus clinical isolates from prosthetic joint infections. And interestingly, the biofilm organism, it does not have to be the phage's optimal host for the phages to be able to break up the biofilms, so which almost completely degrades my makes my later work pointless, why I isolated loads of new bacteriophages. So it'd be good characterization and sequencing work for me. And phage cocktail therapy can be used alongside antibiotics in vitro, we found. I'm sorry about the ambulance outside. And for the next work, we need to finish the genomic annotations of our new bacteriophages that we sequenced with Illumina and MinION, and potentially an animal model to assess the efficacy of our phage cocktail to clear prosthetic joint biofilm infections. Thank you to everyone in the Sagona lab, and especially big thanks to Bethany Pierce, who's been amazing help in this project, a really good MBio student. And if you have any questions after the conference or anything, there's my email and my Twitter. Thank you, everyone.
Thank you, Nathan. I think that was an ideal chocolate torta on which to end the conference.
I thought I'll make it an easier one for everyone, so I thought everyone's brains will be a bit tired.
Oh, you brought lots of things together. It's great. Are there any questions for Nathan about his staff pages or his talk? Because I can maybe ask him. Actually, someone else is asking him first. Yep, go for it.
Hi. So I just wanted to ask that generally biofilms are, if they're more mature, then it becomes more resistant to phage. So have you tried different maturity levels? Would you?
No, that would be an interesting one to do. It's definitely something I would look into next. Definitely.
Mm-hmm. All right, yeah, that's
a good point. It's a good point.
Yeah, thank you.
I wouldn't dismiss the fact that you've isolated all those new phages, because even though the Staph K worked in the biofilm, presumably in an infection, you've not necessarily, you want to try and stop it before it becomes a biofilm. So yeah, I wouldn't be so hard on that interpretation. It's quite scary that I mean, I need to have seen that observation before, that antibiotics really promote a biofilm.
Really inflame.
So how is that happening then? So are the consultants routinely putting biofilm, or the clinicians? Do they routinely put antibiotics into the prosthetic joint before they put it into the?
Oh yes, yeah, that's routine. That's routine during the surgery. We do a massive wash with really high concentration antibiotic, so that might help. But again, they'll prescribe it they'll prescribe antibiotics after the surgery, which again, if they get the infection though, it might cause more damage. If the infection manages to establish.
Yeah. Okay. Well, thank you very much. And as anybody can also, as you said, contact you for more questions.
Thank you.
Officially end the scientific content now. Thank you very much.